Hello.
Hi.
How are you?
Good. How are you?
Doing great. Can you remind me how to pronounce your guys' names just so I say it correctly?
We were just having that discussion the other day with her. There is Deborah and it's Requesens.
Requesens.
Yeah.
Like
It's like rec essence.
Requesens.
Yeah.
Okay.
Yeah. If you did it by rec and then essence, people kind of seems to.
Okay.
Anyway, but I don't care. I truly don't care. I've been pronounced in so many different ways that it doesn't matter.
I feel that the same with my name as well.
How do you pronounce your first name?
My name's pronounced Kanoy.
Kanoy. Okay. Good to know.
Yeah, of course. And then
Yes.
Hervé.
Yeah, that's right. Hervé Tiriac.
Hervé Tiriac.
Yeah.
Okay.
But I think when you're a foreigner, you're just so used to being called anything, doesn't matter.
No worries at all. A quick question for the both of you before everything gets started. When the attendees have a question, they're going to be asked to place their question within the chat box. Would you like me to read the questions off for them, or would you like me to pick a question, unmute that person, and have them be able to ask you the question? What do you prefer?
I think if you read them, it makes it more uniform.
Okay. Perfect. I can definitely do that.
It's like I've been watching the other talks this morning, and I think that's what the moderators have been doing that, and it seems to be working fine.
Okay. Yeah. Perfect. So we'll do that then. Basically, whenever, how is the structure of this chat going to go for you guys?
So I was hoping that Hervé would come here early. So I was looking at those questions, Hervé, and I think like you probably have more experience in anything that has to do with organoids and advanced cell models past the iPSCs. So I think, I mean, if I can chime in on whatever we're doing, but I think that's probably more your.
Yeah, I'm happy. Yeah, I'm happy to take whatever question.
Okay, I think I have more experience on what's the contact with the advocacy groups and the patients themselves, because I do a lot of that side. But I think you, and we only work with 2D cell lines. We're just starting with organoids, so I don't want to speak about something that you have way more experience. Also, the last time, and then I saw that it wasn't several times on these questions, they keep asking about mouse models, and I have no experience with mice.
I can speak about mouse models as well. I have some experience with those for sure.
Okay, I always say I did plant science in college because I didn't want to touch the mice.
Yeah, I'm not a huge fan of mouse work just because of the animal component of it. But I've done quite a bit of it.
Okay, that's great.
I try to avoid it if possible, but I can definitely speak about it.
Okay, that's awesome. Yeah, all the other questions regarding, I think we can all share it, but all the things that has to do with what are the most things that are misunderstood between investigators and researchers, things like that. I have a lot of experience on that side. Yeah. I was just thinking that I know very basic about mouse models. And the other question that I think it might come up, and I hope that you have some more insight than I do, it's why go with one model over another, depending on the disease. And I wanted to hear your thoughts before we even start on this.
Yeah, I mean, my thoughts are very, there's no right answer. It's ideally you use multiple models because every model is going to have something good about it and something bad about it. So when you think about a cell line versus a mouse model, the cell line, you can do many more things much faster. But unfortunately, the cell line is not going to have any immune system. It's not going to have a stroma. It's not going to have this physiological important environment that the mouse is going to have. So ideally, you do everything in at least two models to try to take advantage of the specific model's advantages while also mediating the weaknesses.
Yeah, I was thinking as well as I've heard and I've worked from far away with mouse models that did not recapitulate the disease at all. Like even though we have the same mutation, we didn't see the same effect. So I guess that's, I guess, so I know that yeah, I get asked that a lot. Do you do mice? Do you do mouse models? And we obviously
Yeah.
don't. I don't, like you say. I was watching this morning, they were talking about the organ in a chip, or the,
Yes.
and I was just like, yeah, if we could just keep animals all together, would that be great?
Yeah. Yeah. And I mean, those organ on a chip things are great, but the problem is they're still very difficult to use. So that's a negative. They take a lot of cells, which is not easy to do for like every patient. So you can have like a model that always works, but it's always the same cell. So it's not patient specific, for example.
Yeah.
So there's a lot of issues with those too. Yeah, we have a lot more work.
I know. I mean, I don't even remember how far away I first heard the NCATs talking about this. I swear to God, it must have been like 10 years ago at least, on it was a NORD conference, on one of those where they actually, I think it was him as well, by the way, he talked about this same organ in a chip, and we're still talking about him. And yeah, I think it's not, we haven't won that battle yet.
So I have the questions in front of me now, and I was wondering remind me, Kanoe? Yeah, okay. Kanoe, so we're gonna let the attendants ask their questions first, and if they run out of questions, or if they don't ask any questions, I guess you're gonna pick these questions kind of randomly or in order, or how do you want to do it?
I'm gonna ask them in the order, because that's kind of the way Maureen made them. Perfect. As like level of importance. So I'll make my way down that list as we get through them.
Sounds good. Sounds good.
Awesome. Yeah, I'm hoping that people will be. So you're gonna introduce us a little bit, and then we're gonna give ourselves I don't have any slides either. I don't know if Hervé has any lines. Okay.
That's perfectly fine. So what I'll do is I'll give you both a very brief intro, just your name, where you guys are from, like work-wise, and then I'll give it over to both of you guys to further introduce yourselves, give more information on your background.
Okay.
And then from there, once you guys are done introducing yourselves, is there any other things you want to talk about, or do you just want me to kind of get things started with some questions to kind of get the discussion going?
Yeah.
Yeah.
Okay.
Just get started.
Okay,
Yeah,
perfect. So once you guys are done, if we don't have any questions already in the box, I'll just start at the top of the list of questions.
Okay.
And then hopefully through that, we'll start getting some more from the attendees, and we'll go from there.
Yeah.
Sounds
good.
Cool. All right, so we got 7 minutes to just chill. Wait.
How are things in California? How's the weather?
You know, the weather has been pretty good lately. It's gotten really hot the last few days, at least in San Diego.
Yeah, it's been hot over here in Orange County as well. So
It's getting even hot here. I'm in Philadelphia, and we've been in the 80s for the last few days, which is weird for us.
Oh,
yeah.
Wow.
It's rain today, but it's very, very humid up here as well.
Yeah. We were we decided to put an AC, which normally we don't need, because in San Diego, if you live close enough to the water, you don't get the strong heat. But we decided, since we're going to be working from home for like so many months, might as well upgrade. And it was a great decision, because let me tell you, working from home when it's hot is not fun.
I can imagine. I mean, it's funny that the amount of home improvements that is going on in people's houses because of being home is like, I didn't have an office before. Like I didn't have an office before I started this. And like after 2 weeks, when I realized that I wasn't going back to the office, I said, I don't care. We're taking that guest room and making it a real office. I cannot continue to work from my couch. It's not happening anymore.
Yeah, it was fine before for like a few hours of work here and there, but you need something professional now.
Yeah, I was getting backaches just working out of a chair, of a couch. So it's a lot of things. A lot of people are you guys going by? Are you closed? Is the university closed?
No, so I'm actually at the university right now. I'm in my
I
office.
can see that, but I mean, you guys are not open to.
No, they reopened. They reopened this month in what they call a ramp up phase. So the University of California has like 4 phases: red, orange, yellow, and green. Red was lockdown. Now we're in orange, which is like 30% occupancy and shifts, and monitoring. You need to have a mask, but I mean, I'm not going to wear the mask in the office. When when things de-escalate a little bit more, it's going to be 50% occupancy, and then we go to 100% after that.
Okay.
So it's going to take a few months, hopefully not more than that, before we can go back to green. But
Yeah.
we could be an orange/yellow for 6 months plus. I don't know.
Yeah. We never closed because our labs are deemed essential, and we never stop production. But those who work from home are requested to. We only go one day a week. So tomorrow you'll see a different background, because tomorrow I'm gonna be at the office all day. But yeah, it's been so. Yeah, but actually our president doesn't want to let us back on fully until a vaccine is ready. So we might be doing this for a long time.
I think that's what's going to happen here too. I'm worried that we're going to be stuck in this semi-open phase.
Yeah, we have 30% capacity right now. It's what we are allowed. So if the labs are there, then the people in offices can only go once a week or something. And we also get tested once a week. So we go to the office mainly to get we develop actually a saliva test that is very it's easy to, because you just literally spit on a tube.
Yeah.
We get tested every week, so
that's great. Yeah.
My whole neighborhood is very happy with it. I think they feel like if I don't have it, nobody else does. Kind of thing. But yeah, it's very good actually. We work with a hospital, so we develop it for the hospital, and we do it for our own internal testing. So that's kind of cool.
Yeah. The hospital here, the UC San Diego Hospital, developed their own test too. I don't know exactly what type of test it is, but it's a priority scheme. So like clinicians are highest priority, patients obviously are the highest priority, and then researchers on the surrounding sites are the next priority. So I'm on a wait list for whenever they get to my name to get the first time test. And it's definitely not going to be once a week. They want to get it to once a month once they have enough throughput.
I think the saliva one is great. And I think even, we don't do that, but we're talking even doing about pooling, which is what other countries have been doing. And I think that actually will work, because we haven't had a single test positive, and we're testing 90 people a week. So if we had done pooling all this time, it would have saved so much material and time. And then you only retest the whole week, But I think that probably will move into the pooling.
Even if you do pools of tens, I mean, it would save you a lot.
Yeah, it will definitely. And then if it happens, then one comes up and then you do it again. But that's very rare. It wouldn't have never happened in the last 3 months for us since we developed the testing of plants.
That's awesome.
But yeah, so I consider myself lucky in that sense. Now, hopefully, do we know if we have people register for because we didn't know before if we were going to have a big group of people attending.
Yeah, so currently we have one person already in the waiting room, 2 people in the waiting room. So I believe the sessions are just getting out right now, so we should start seeing some more people come. Okay. But I do know we had a handful of people pre-register ahead of time. I don't know the exact number off the top of my head for this specific one.
Okay. But
I know we did have a couple people who registered ahead of time. So
that's good.
Yeah. So we're just gonna, once it hits 11:30, we'll wait a little bit for some more people to start gathering, and then I'll let them in, and then I'll make sure that they stay muted. And as soon as we start seeing a good majority of the people start rolling in, we'll get things started.
Sounds good, no rush.
Great.
I'm sure it'll be easier tomorrow after people get more used to it today.
Yeah. I'm gonna message it.
So far, the platform seems to be working great, by the way.
Oh, that's good to hear. I'm glad you're having a good experience with it so far.
Yeah. In spite of how sad it is to do everything virtually.
Yeah. Well, it's going to be, it's unfortunately the new reality, right? It's interesting to see that they have like a virtual conference center, which is kind of cool. It's better than just a bunch of links.
It's a virtual reality, exactly.
Yeah.
Yeah, it's definitely really cool to see the different options that they have now for virtual events. So,
although I cannot wait to go to a real conference.
Oh yeah, we definitely cannot wait either. Really looking forward to it.
So, Bernard, do you work for Global Genes?
Yes, I'm the event coordinator.
Okay.
I've been with Global Genes for about a year.
How long have you been with them?
Yeah, for almost about a year and a half now.
Were you there last year for the Global Summit?
I was, yes.
Okay. Yeah, I was in one of the panels last year.
Yeah, I know your name sounded very familiar.
Yeah.
And Corey does a lot of like exhibiting with
Yeah.
us too.
Yeah.
So
yeah, we've done both. But yeah, last year I spoke at a advocate panel.
perfect.
Yeah. Do we know if we're going to have that meeting this year? Probably we're not talking about it again for the summit.
For the summit, we did change it to a virtual style meeting. It's gonna be, yeah, unfortunately we had to make that call, but it's gonna be from September 14th through the 25th. We have some more information about it online, and then we're gonna be releasing more like with a specific agenda within the next few weeks.
Oh, that's sad. I love that meeting, and I love going to California, and especially since we moved back to San Diego. I was so excited to go back. I love that town. I love you guys.
I know, we're really bummed, but I'm gonna start letting some people in right now.
Perfect.
We currently have 3. So I'm gonna get them started coming in right now. Let me do this. Hello, everyone. Welcome. So we are gonna wait just a few more moments for some more people to join us today. So just give us one couple more moments. All right, we got some more people coming in. We'll get them in. All right, and then for those of you just joining us, we're just giving a couple more moments for some more people to join in. Do keep yourself on mute, please, for right now. All right. Okay, great. So Deborah and Hervé, I'm going to unmute you right now. Where are you? Okay, perfect. All right, so we'll just go ahead and get started, and as more people join, I'll just let them on in. Good afternoon, everyone, and welcome to the Rare Round session today. So we are very excited for our upcoming discussion on cell models in rare disease research. So before we get things kicked off, I do want to go over a couple of brief housekeeping notes. So first, all attendees are currently muted by myself to reduce any interruptions throughout the session. We do encourage you to please use the chat function in the Zoom window for any questions you may have throughout the sessions, and then I myself will present any questions you have to the speakers to answer. To open the chat, please click on the chat button located on the meetings control panel at the bottom of your screen near the mute icon. So if there are also any questions that we are unable to address, we will try our best to get back to you after the program to answer those questions. But yes, also in the interest of making this feel more connected for this discussion, if you would like to turn on your video, please feel free to do so at your comfort, of course. So let me just admit a few more people into the room real quick. All right, here we come. Okay, so finally, we encourage you all to participate as active listeners and take advantage of this opportunity to hear from leaders and experts here and ask any questions you may have of them. This program is really your time to learn and to find answers on critical challenges in rare disease research. So without further ado, I am now pleased to welcome our discussion leaders for today. First, we have Deborah Requesens from Coriell Institute. And then we also have Hervé Tiriac from the University of California, San Diego. They both will each share a more complex or a more complete background about themselves, and then we will dive into the conversation from there. So I'm going to kick it off to Deborah to introduce yourselves.
Good afternoon and good morning for those on the West Coast. My name is Deborah Requesens. I am the principal investigator of the NIGMS repository. This is a repository of cell lines and DNA based on rare disease patients and their families. I'm happy to answer any questions. We are, by the way, located at the Coriell Institute, which is a nonprofit located in Camden, New Jersey, just outside Philadelphia. I'm happy to answer any questions that you might have regarding the establishment of cell lines and how the repository works.
Perfect, thank you, Deborah. And then Hervé, if you would like to introduce yourself.
Hi, everybody. I'm a research scientist at the University of California, San Diego. I work in the Department of Surgery here as a researcher, where I work with a lot of the clinicians to establish patient-derived models. So specifically, I've been working on organoid models of cancer for many years now. I did my postdoc on the East Coast before I came over here to start this little laboratory effort. And we use these patient models mostly from pancreatic cancer patients, but as well as other types of cancers to understand how patients will respond to chemotherapy and other standard of care treatments, and to try to see if we can predict the outcome and identify treatments that will be beneficial for a patient versus treatments that are not going to have any great effect and are going to lead to toxicity.
Awesome, great, thank you both. So what we're going to do now is I'm going to start by asking both Deborah and Hervé some questions. And as you all attendees think of any questions, please feel free to write it in that chat box, and I will then ask them to our discussion leaders over here. So we'll start off with our first question. What are the advantages and limitations of various types of models?
So I can start on that. So there's many different types of cellular models that you can use for research, anywhere going from the very reliable and very common two-dimensional monolayer cell lines to the more advanced genetically engineered mouse models of disease. And each model has benefits and drawbacks, and they have to be applied in a thoughtful manner to the question that is being asked. So for example, if you want to screen hundreds of thousands of chemical compounds to find a new drug for a specific disease, the ideal model to start with would be a monolayer cell line, just because you can make those models, you can amplify them really quickly, and you can screen them using robotics very easily. So that is an achievable experiment. But if you want to understand how the immune system will respond to immunotherapy and go in and kill a cancer cell, then you cannot use a monolayer cell line where all you have is the cancer cells, for example, or the disease cells. In that case, you have to turn to an animal model where you have both the disease cells as well as the immune component. And typically, at least for cancer research, those are genetically engineered mouse models, which are, there's no human cells in that system. It's a pure mouse system where you have both cells that are engineered to recapitulate the hallmarks of the disease that you're studying, as well as the full immune and stromal complement, so you can ask complex questions on the microenvironment. And then in between those two extremes, you have models like the organoid, iPS cells, or organs on a chip, which add in multiple cell types, or they become more complex by growing in 3D. And each of those have their own advantages and drawbacks, which we can discuss in detail if you have questions.
I just wanted to add, I couldn't say more about that. The only thing I wanted to ask, even at the very simple level of a 2D layer, monolayer of cell lines, we even that have different types. We have what we call LCL, so lymphoblastoid cell lines, which is just immortalized cell lines that don't really represent anything, but you even have fibroblasts, which is something that the researchers use that, and that will really depend on the disease, and some diseases that are more based on muscles, and it's necessary to look at the fibroblast, then we do that. So even there's differences at the most basic level.
Okay, great, thank you. It looks like we got a question over here. So are there feasible ways for anti-cancer high/medium throughput screening in which tumor microenvironment is preserved?
Yeah, so I would say medium throughput for sure, maybe even low throughput. It kind of depends on how you define these different thresholds. So if you're talking about hundreds of compounds, you can do those screens on multicellular models like organoid co-cultures or organ on a chip. It just requires a lot of investment upfront in terms of effort to grow up all the cells and make up all the models that are going to be screened for this. And typically, because of the complexity of the system, the automation is not quite the same as the high throughput systems. But those types of screens, which I would call more complex, are coming online now, and there's papers for different diseases being published on this. So it's definitely an approach that's going to be that is being used right now, and that's going to become more and more popular as people adapt their machinery to be able to take advantage of these complex models.
Great, thank you so much. All right, so we'll move on to our next question. What are the ballpark costs of developing and maintaining cell models, and also of sharing the models with researchers as well?
I can go on that one. So it really depends who you're working with and what is the purpose of your research. In my particular case, I work with a repository that is funded by the NIH. So the cost of establishing and developing a cell line, for example, it's free of charge for the patient, and it's literally at cost for the researchers. The same thing, so the distribution, and that's more of the sharing, and the sharing with researchers is really at cost. We do provide, and many companies do provide, establishments of cell lines, and the cost of a regular cell line, and I'm not talking about an induced pluripotent stem cell, I'm talking just an LCL or a fibroblast, is negligent. I think the biggest issue that people have to remember is that this not only comes, you establish this cell line, but what do you do with it? So you have to think about all the quality control parameters, and then you have to think about storage. So you need a biobank who storage the cell lines at the appropriate conditions, and then you have to think about the sharing, and that takes a lot of, believe it or not, that's actually the highest cost. It's just managing the relationship about the sharing the cell line. So the cost is not just of developing. Again, establishing is just a few $100s, or even a few $1,000 if we're talking about an iPSC. But the problem is what comes after that. So it depends who you're working with.
Yeah, and I'll second that. It's entirely dependent on the type of work that you are planning, because in terms of materials, it's negligible compared to the work hours required to do something with that model. So definitely depends on what you want to do with the model.
Okay, great. Thank you so much. So for the next question we have, what are some new advances in tools or technologies that can facilitate development of cell models?
Yeah, let you start.
Sure. So I'll speak specifically about organoids since that's my specialty. But keep in mind that there's multiple different types of models out there that are sort of up and coming. So organoid models have been developed by many laboratories. Most people these days, when they talk about organoid, they're talking about the Hans Cleaver's methods of developing these organoids, but that's not the only method available. And that's a method that takes advantage of specific growth factors to isolate and expand the epithelium of different diseases. That can be both cancer and non-cancer type of tissues. The advantage of organoid models is you can also grow normal, healthy, non-disease control organoids in the same conditions that you can grow the disease, which allows you to do really nice experiments that have a normal control and a disease control, which has been difficult to do for many other types of models. These methods are actually quite reliable, and it's not too difficult to establish it in your laboratory. It does bring up a set of new challenges because now you're working in 3-dimension compared to the monolayer cell lines that you were using before. So the costs are a little bit higher, and the technique is a little bit different, and it takes a little bit longer to work with these models, but it's definitely achievable and has been transferred from laboratory to laboratory pretty easily, as long as the investment in terms of learning and getting the reagents specific to the cultures have been followed. So that's one method. The one nice thing about organoids is we're making them on a per patient basis. So we're now generating large cohorts of organoids for specific diseases so we can study the phenotypic heterogeneity of the population of interest. So for pancreatic cancer, we're now making hundreds of organoids, so we can identify those small percent of patients that will respond to a very specific drug that only they will respond to, compared to like the 98% of the patients that don't respond. And that was difficult to achieve with previous models just because of the throughput was not there. So that's one example.
I like to add that I think one of the newest, and it's not that new anymore, but the new tools that are very useful now in the models is the gene editing using the CRISPR-Cas technology. This is a very, I'm sure everybody that is in this world kind of has heard about this, but it's just a simple way of adding a mutation or correcting a mutation in a cell line, and they're very useful for creating what we call isogenic controls, which is taking the cell model, the cell line from an individual and creating exactly the same model without the mutation. So this is something that we're using a lot in our laboratories and a lot of people around the world. I was thinking about that, and also I was thinking about how easily affordable and fast it is now to differentiate stem cells. So that's something also very useful when the disease can only be studied on the real tissue that is affected. So we, based on iPSCs, now we can differentiate them mainly into neurons or cardiac cells. That's mainly what we work on, but I know that we're thinking of pancreatic cells is something that are being developed from this iPSC. So those are all techniques that are becoming current in labs, and the needs, the investment is not that high.
Thank you. And then for those of you who have just joined us, if you have any questions at any time, please feel free to leave those questions in the chat box, and I'll be able to ask those for you. Moving on to our next question now. What are some key...
There's a question.
Oh, here we go. So speaking from a patient advocacy standpoint, how can we be most helpful providing biomaterials? We are building a biobank and have access to blood and tissue samples. We are trying to improve access for all researchers. From Kristen Anthony.
So I'm happy to answer that. This is kind of my bread and butter. That I say this so many times a week, sometimes. So the advocacy groups and the patients themselves can definitely be part of the research. You can reach out, you can create your own biobank, of course, and I think that this is, as many people will understand, then this is costly. So then you can work. I'm happy to answer then after that any questions, since we work for the rare disease biobank, and we work with many of you. The way that we work is that we provide information. You can let your patients know. We work, I give presentations at different conferences, but we also put things on their website. So the information is out there, and once that happens, it's up to the patients to be present, to actually act for their patient advocacy groups and contact a biobank such as ours, and just give access to their samples. I think that you can participate in research very easily, especially, and I think there's a question about that later. I don't know if we'll get there, but especially if you believe in that the more people that have access to your sample, the better your chances of obtaining a treatment or a cure. If you are of that idea, which is my goal, then you should definitely look into working with a large biobank that has free access to your samples.
Mm-hmm. And if I can just build upon that, it's important, if you're building the biobank, that you consult with researchers that you want to partner with in the future. Because, for example, I've been approached by different people that have asked if we can generate organoids from tissue samples that were pre-existing in a biobank. Unfortunately, we need very specific preservation methods to maintain the cells frozen before we can generate an organoid. So they still have to be viable before you can make the model. So the biobank has to be up to a certain spec if you want the research to then be doing something very specific with it. That's one comment. And then the other comment is, once you become very famous and a lot of researchers want to work with you, it's important to have some sort of path to prioritize the projects, because human samples are extremely, extremely valuable and limited, unfortunately. So it's important to prioritize them somehow, and it's helpful to have an advisory board of scientists and clinicians and patients that can sort of together make a decision based on what is feasible, what is most likely to succeed, and what the patients really want to accomplish.
Gotcha, thank you. And then we also have a question from Charles Hoffman. He asked, are the screens only using drugs with known targets? Are these ever done with compound libraries of unknown activity?
Yeah, so I can follow up on that. I was part of a very nice group of scientists and researchers that met up at the NIH a couple of years ago to discuss natural product research. So there's an entire section of the NIH, I forget the name of it, that deals with natural products. And those are basically, molecules or chemicals that come out of different plants and different microorganisms. And they've been categorized or cataloged in large chemical libraries. We're talking about hundreds of thousands of compounds. And those compounds don't have most of them don't have known targets. They don't have known activities. So they're definitely being used in research to identify maybe a new cure for a disease that previously didn't have a cure. The one issue with it is a lot of natural products are complex, meaning that it's not just one molecule that holds the activity, it's the combination of multiple molecules in the plant, for example, that will have a very specific activity. So it becomes a little tricky to do the research, because then you need to balance out the different chemicals. But it's definitely possible to do screens with these compound libraries of a known activity. You just have to do a lot of research once you identified hits to validate them, to make sure that they're actually doing what you think they're doing, and that the chemical that you think is active is actually the one doing the activity.
Gotcha. Thank you so much. And then we have another question from Tracy Marie Stewart. So she asked, can you comment on IRB requirements for developing patient-derived cell lines?
I can answer for that. So there's two ways of doing this. You can either work with an investigator working on your disease who should have its own IRB at their institution. And these IRB are, they vary from a protocol to protocol, from institution to institution. In our particular case, we also have our own IRB that has been approved for the NGMS repository itself. It's part of the Korean Institute IRB, but we have our own IRB protocol for it. And in our case, we can also work under the IRB. They all are different. They have different parameters that you have to follow. You have to ask, like in our case, we don't allow commercialization of our products, for example. We're very strict about our distribution and our sharing and the way the samples can be used further down. In this, you have to make sure that you look also at the informed consent form that you're signing in, that is part of the IRB, but that's usually what you guys can see, right? Because the patients are very rarely able to look in details at the IRB protocol, but you can look at the informed consent, and you should, and very carefully before you sign anything. And in here, you'll have the details that are, explain more details on the IRB, but they will explain you what are the possible uses of your samples and what are the conditions and what kind of data could be shared and in which way.
Thank you. Next up, we have another question from Yessa. So we have a biobank in Italy managed through telethon, but the USA patients are underrepresented due to barriers to travel to international conferences and difficulties in shipping samples. Is it better to keep biobank all in one place, or should we develop a separate biobank here in the States?
Um, you know, and I would actually go with the one centralized location for all your samples, just because it happens often that we have duplication of submissions. People and patients will, and investigators will actually share their samples with different biobanks, and sometimes we are unaware of what else samples are present, and that happens a lot. And well, we do a lot of testing on identity, in identity, and we make sure that the samples are not duplicated, but that does happen. So I tend to go to the one biobank, but I do understand the difficulties of shipping samples. This shouldn't be an issue, at least in our particular case. We receive samples from all over the world. So we don't have any barriers regarding we ship our kits to anywhere in the world and they come back to us and we establish the bank. So perhaps in Italy, you could be able to do something similar.
Great. Thank
Yeah,
you.
I don't have a lot to add to that. The one effort that I know is one called the Human Models Initiative, that is spearheaded by the NIH, which is trying to generate organoid models and cellular models for many different diseases in the cancer space. And they're taking models from the US as well as Europe. But I know that it was a little tricky to have the tissue shipped because of IRB issues, have the tissue shipped to the US for the model generation. So instead, the model generation is happening in the country where the tissue is collected, and then the models are shipped to the NIH. Which is less problematic because at that point the tissue is the models are de-identified, so that avoids a lot of the IRB issues with the tissue itself. So,
Got it. Thank you. Thank you for that question. Next up, we have this question. So, what are some key misunderstandings on this subject, and what are some common missteps by advocacy groups and by researchers?
I have a little bit, and I'm sure we have so much to add. I think there are no misunderstanding. I think they're just, the knowledge is not fully there, and I think that's why we do things like this. So is the difference between what is a cell line. Something that I get often is the misunderstanding of thinking that when an LCL or a fibrous cell line is the same thing an iPSC, just because they have cell line at the end of the word. They're very different models. That's something that just, but because again, this is no misunderstanding. This is just a question of knowledge. I think the most common missteps by groups, I think it's a lack of communication sometimes with the investigators. So a lot of times samples are given to investigators that are going to use them for their own research, and then they won't share them. And I know David and I have talked about this a little bit before. It's just the sharing is a huge deal. So when you sign a release of the submission form, make sure that you understand where your samples are going. Would you be able to move them to another location if you wish so? Would you obtain any results from the studies doing on the cell lines? So I think most of the missteps or misunderstanding comes from miscommunication.
yeah, I'll give a personal example. We published a paper a couple of years ago stating that we could derive organoids from pancreatic cancer patients, and that these organoids had a predictive power in selecting therapy for the patient from which they were derived. And the entire work was done with de-identified materials. So we had de-identified clinical records and de-identified tissue to make the organoids. And of course, when we published, there's a lot of PR happening at the institute level and also at the funding agency that provided the funds for the research. And so the research is being disseminated in little news articles. And we started receiving phone calls and emails from patients asking if we could start making organoids from their tumors to try to help their clinical care. But then the issue and the confusion there is our work is in the research phase, not in the clinical setting phase. We haven't proven in a clinical trial, in a large clinical trial, that our method is actually predictive. We still need to do that. So we don't have all the regulatory requirements to be able to just do this work now for every patient. So it's a little tough to sort of navigate the space of doing translational medicine, publishing it, advertising that you've done it, and then saying, well, I can't help you because, I'm still not quite there. I need to go a little further. There is, however, a little space where patient and advocates can fit in this research phase, but it is really hard to navigate, and it does go back to your RRB question on how can patients actually participate in the research involving their models when most of the time the RRB states, at least from my perspective, most of the time the RRB states that we're going to get the identified tissue. So I automatically don't know, whose tissue I'm working with, and I cannot re-identify it by design. So that is something that both researchers and patients should probably be discussing, and at some point involving the IRB at different institutions to try to craft new IRB regulations for these. And some of that could also come from the NIH.
I think that's one of the biggest things, and advocates are always asking what can they do and what can they be pushing for, and that is something that I believe it has to come from the advocates and from the patients themselves. Push for this to change. Traditionally, investigators have not wanted to know even. I mean, and I'm not saying, I think I'm talking maybe the next, in the past. In the past, it's been one of those, they prefer to obtain a no identified sample, so they don't have so the patients cannot go to you, like you're saying, be like, so what happened to my sample? Did you get a result? Can I do something about it? So, but I believe that should change. And I believe it is changing. And I think somebody this morning was talking about, I think somebody from the Global Gene saying that in Europe, that is changing a little bit already, where we're making the patients more aware of the results. And I believe that's something that should change here. But I believe that the patients advocates and the patients themselves have to be the ones helping us change this.
Great, that's great insight. All right, so our next question we have is going to be, what are some of the strengths of different types of cell models for different categories of rare disease?
I mean, it really depends on the cellular model you're talking about. So I guess a big strength would be if the cellular model recapitulates the entire disease phenotype. So for a rare disease, maybe every patient has the exact same mutation, in which case maybe 1 or 2 cell lines is enough to be able to study the entire disease. But if it's a little bit more complex and there's like a dozen mutations that lead to the same disease, then you need at least a dozen cell lines, and ideally multiple replicates of the same mutation in different cell lines to really have something where you can study the entire disease for the entire cohort of patients that have this disease. So that's maybe number one, is to be able to generate models for the entire phenotypic diversity of the disease. And I guess another consideration, going back to the first question, would be to have multiple types of models. So having a cell line and having a mouse model would be nice, because then you can do multiple types of experiments. The ones in the cells that are high throughput, and the ones in the animals that are more microenvironment focused and physiologically relevant.
Okay. Thank you.
More to ask that.
All right. So the next question we have, so at what stage in research should you invest in developing a cell model?
I feel like I think that goes back to the question that Ray just answered. I think it depends on the cell model that we're talking about, right? If we're gonna start, I always recommend that if you're working with a public biobank, like something funded by the NIH, start right away. I mean, all you have to do is give blood, right? So why wouldn't you start with that? It gives 2 things. It gives the opportunity, so in a few months, to have a cell line that you can use for something else, and also gives the opportunity to obtain a lot of the DNA, which is, we haven't mentioned it here, but DNA is very important for many other type of researches, and the characterization at the genetic level is become so cheap that it's become an important part of any kind of cell model to characterize it at that level. So why wouldn't you make sure that the DNA is available not just for testing, but also not also just for studying, but also for testing, right? All the genetic testing needs your DNA. So for all the reference material organizations that are going to be testing on your disease, they need the DNA to be able to validate their testing. So why wouldn't you start with something like that, that is absolutely free of charge in many of the NIH repositories, and they will be available to researchers. So I will start right away in that level. For something like a mouse model, I think that is something that needs to be happening a little bit later on. And I don't know if you have any insights at one point. Do you think of it that we should we start talking about something more advanced.
No, again, I can speak from personal experience. I was involved in the Caring for Carcinoid Foundation for many years, and they've been having a really tough time in that disease to generate a single relevant model, be it either mouse or cells, just because it's a very difficult disease to model for whatever biological reason. So they've been every year, looking at different research proposals and funding different research proposals to try to generate models. And there's been some, mild success, but it's been very incremental and very slow. And it's been such a high priority for them over the years. And it still hasn't completely succeeded. So it can be very difficult to generate a model for a specific disease sometimes. And sometimes it can be very easy, but it's probably worth investing in that right away as you're trying to start a foundation or an advocacy group, just because the model will enable a lot of research downstream of it. So the sooner you do it, the better you are. And in the absence of a model, like we just said, having tissue and DNA and all that is invaluable. So definitely bank those.
Gotcha. Thank you. And then we have a question for Deborah. So you mentioned that your policy is non-commercial use. Can you clarify what you meant? How does that play out if a company wants to use a cell line for early stage development, obviously looking to possibly make money later down the road?
Sure. So yes, it's a great question because it can be confusing the way when I say non-commercial. So no, so commercial labs can definitely obtain your samples. So a scientist, a researcher is able to commercialize the information and the research results that you got from working on a cell line or DNA from a repository. What you cannot do is take that cell line, that DNA, and put it in a commercial product that will be sold. So which means is that your sample itself will not be sold, but the knowledge obtained from using your sample could be commercialized by a commercial entity. Is that clear?
Okay.
Awesome.
Perfect. All right. So next question. What are some key reasons cell models might fail?
There's many.
Well, I mean, people forget that they're living organisms. It's a living thing. So sometimes they just fail. Sometimes they just don't want to grow. Things are like the age of the patient can influence the way a cell culture thrives or not. A lot of times it depends on the mutation itself. And I'm thinking more of the way that we work on reprogramming iPSCs. Sometimes we cannot. Sometimes there's some chromosomal abnormalities or chromosomal tendencies for the cell lines to not differentiate, differentiate. So it truly, sometimes we don't know, sometimes it has to do with the mutation itself. That's at the level of cell lines and the basic level of a cell line.
Yeah. And sometimes it also has to do with the way you use the model. So there's somewhat infamous examples in the literature of a really cool research result happening with a mouse model where treatment led to tumor decrease, for example. And then you translate this to the clinic in a clinical trial over many, many years. And the clinical trial fails in a really unfortunate way where the patients actually progress on treatment. And then you realize that the mouse trial, in fact, did predict the outcome. Unfortunately, it was not seen because the mouse trial was only 2 weeks, because mice trials are really quick. Whereas in the patient, the treatment went on for months. And initially, yeah, there was a benefit for a couple of weeks upfront. But then after a long time, the treatment actually led to negative effects. And that wasn't observed in the mouse trials because they were too short. So sometimes you also have to kind of, and it's hard because you have to go back to the trial as you're doing as you're planning clinical trials to really answer all these questions. But you have to think about the clinical reality of how these treatments that you discover in models are going to be translated in clinical trials. And you may have to redo studies or further studies to really make sure you're modeling the entire clinical care.
That's good to know. Thank you. So our next question that we have, How do you see cell models addressing complex rare diseases where variants in one gene might be the causal mechanism, but there are several different types of variants? The causal mechanism.
I think that this is the case of, well, first, cell models can address that when we're talking about actually basic cell models, because we can use gene editing to, do different mutations in different parts of the gene. We can actually, we can knock out the whole gene, and that can also be done in mice. So I think the genetic editing of all these models can be a way to address this. I think also the biggest thing that we have with the difference between a patient-derived cell line and a mouse, for example, is that only the patient-derived cell line will tell you about other genes and other variants that might not be in the spotlight when we're talking about that disease. That when you do, you take an animal and you just work on that known variant, then you don't think about the other things that might be causing, having an effect on that main causal gene code. So it's something that, yeah, it happens. I guess is that's when you realize it afterwards, right? Like there's no way to know. Although now, I guess with the whole genome sequence, we can actually look at many other variants in a patient and try to compare to other patients with similar phenotypes, and that's how we found many diseases and how find many of the mutations in these new diseases.
Gotcha.
There's a cool example in the Netherlands of a trial where they're using organoid models from patients with cystic fibrosis, and the mutations that cause cystic fibrosis are really well categorized, and there's several drugs available. Unfortunately, it's unclear which mutations will respond to specific drugs. So they're making organoids for the patients and then treating the organoids with the drugs to pick the drug that's going to actually work. And this is actually a clinical trial in the Netherlands, and my understanding is the insurance company have signed up with the clinical trial to actually provide the drug for free, or not for free, but to cover the cost of the drug for the lifetime of the patient once the organoid predicts the response. However, if the organoid fails to predict response, the patient, one, doesn't get the drug, and the insurance company doesn't cover drugs if the patient decides to get the drugs. So there's definitely ways to sort of model different variation of a mechanism in a model and to find good drugs for it, for example. But yeah, if you start implementing that in the clinic, once precision medicine gets going, it may lead to some interesting results with insurance companies.
Gotcha.
Slight tangent.
Thank you. All right. So next up, what are some of the key caveats associated with cell models?
I feel like we've done nothing but answer that question already.
You want me to answer a different one or ask a different one?
Sure.
Sure.
All right, we'll move on. So what kinds of skills and expertise should advocates or researchers look for when funding or collaborating with a researcher?
I can go and start with that one. I think the main skill that you have to find is I think it's an open communication, and I go back to that before an advocate. I start to working with a researcher or thinking who they're going to collaborate with. I think it's important to find out exactly what's going to happen to the sample, the ownership of the samples, the rights that you let go of or maintain over your sample once the model is established. I think that it's important to look at what kind of quality control parameters will be established on your cell lines and your cell models. I think these things are not described openly because it was believed in the past that patients didn't know, and we operated under this system that the doctor knew better. And I think that's completely changed. I work with many of you guys, and you know more than the doctors. So I think that's totally changed. So you have all the right before you collaborate with a researcher to find out exactly what's going to happen to my sample. What kind of quality control do you do on it? Who are you collaborating with, and under what parameters, and what are the chances of me being able to change it to somewhere else? What are the uses that you're gonna, read in detail your informed consent form. If you have a chance to look at the IRB, do that. So that's the kind of things that I will start with before I collaborate with a researcher.
Great. All right. Okay, so we got 15 minutes left, everyone. So please, if you have any questions, go ahead and place that in the chat box. We do have another question over here from Charles. So traditionally, medicinal chemistry is guided by enzyme assays to enhance targets specificity. Sorry, can't pronounce words right now. What do you think of the idea of using tissue selectivity instead when the target is actually a protein family that may be expressed with different profiles in different tissues?
It's a great idea. Yeah, it's a lovely idea, and it's something that I think a lot of researchers are trying to take advantage of. The only issue is human physiology or animal physiology is complex, and there is a circuitry there that typically, starts in the liver, and so a lot of the enzymes that will modify the drugs are in the liver. So there is, in pancreatic cancer, there's a few prodrugs that are modified by the liver before they become active. So it's being used already, but the strategy can be a little difficult just because there's a path that these drugs have to go through depending on the delivery method. But if you can achieve it, then increasing specificity is really cool in disease. So that's a great idea.
Did that answer your question? Perfect. All right. So next up, we have what kinds of controls should be considered when developing or testing cell models?
I think the most, there are several controls that we consider when we have a cell model. I think that the easiest one, first of all, is working in trios. So working with non-affected family members. Those are very interesting and also in different type of studies. So we work with both parents, unaffected parents, and unaffected siblings. But I think more and more people are working with isogenic controls, which is the use of gene editing to take the patient-derived model and then taking away the disease, if you will, and making it a healthy control that has exactly the same genetic background as the patient-derived line.
Yeah,
that's mainly what it is, right? Yeah. I mean, the other thing that we have, and I think it's a little bit more complex, are age controls. And then that we have that specifically with the iPSCs, where we're thinking about age controls, but those are more ethically complicated because it's complicated to just have iPSCs to be used in research from a healthy patient that is a child, that is a minor. So, but that's another control that we see a lot in the research.
Yeah, and in addition to age, gender balance in the study is really important. And that's one that every time you submit a grant to the NIH, they make sure that you have that language in there because otherwise you may miss really important gender-specific phenotypes, which, are rare, but they occur.
Good to know. All right. So next up, how can we ensure licenses are not hindering access to cell models by all stakeholders, including other academic groups and researchers in the industry?
I can tell you in our personal experience, most of the licenses, we work specifically with licenses for reprogramming iPSCs. If they are used for research only purposes, then you don't need to do much more than that. It becomes an issue when you are going to commercialize the product which is a different thing. We are finding, and I don't know if it has the same but issues right now with things like gene editing, which is what I've mentioned several times. We have a lot of issues with that particular license. The CRISPR-Cas9 now belongs to the Broad Institute, and every user of this technology needs a license. So yes, so you work with it. We're working with a legal department. We're trying to make sure that we comply with the regulations. We are spending the money necessary to be able to operate under this license. You talk to the institutes that have the license. You make sure that you explain if you're doing only research only, then you should be okay. But it's a discussion.
Okay, thank you. And then we have another question for you, Deborah. So not sure if this was discussed, but are the cell lines always de-identified? I am thinking integration with registries and with data science, where genome phenotype information is important.
So yes and yes. So cell lines are de-identified, but we do work with many groups that have their own registry. So as long as we work in a and it's semi-de-identified in the sense that whoever is obtaining the cell line will not be able to obtain that kind of information, but the group in charge of the registry will be able to have a code at least. They will be able to go back and forth between the phenotype and the genotype information that you get and what's presented in the repository. So yes and yes. Was that clear?
Yes, perfect. All right. So next up, how can we ensure that cell lines are used in a way that patients are comfortable with?
Well, I mean, it starts with the consent form. You know, the patient has to sign it. And if the language that the patient wants to see is not in there, then probably they should have a discussion or not sign the form. I don't think you can add language on the fly. I think it has to be all approved by the IRB before it's put into the document. So yeah, if you're not happy with the language of the consent form, don't sign it. If you have the luxury of time and you can work with the IRB to generate a consent form that you're happy with, then I would recommend you do that.
Yeah, read every word in your informed consent. If you also have a submission form, like we have both an informed consent form and a submission form, make sure that you read both of them in detail.
Yeah, we try very hard not to make them too long, and there's somebody explaining it, every part of it. So
yeah, and we work constantly on simplifying the language. We even have a special one for kids 15 and to 18, so it's even more simple. So we do make sure that it's at a reading level for non-scientists to understand. But yes, and if you don't understand, I recommend that you ask either a genetic counselor or you ask the group that you're working with to clarify any point that you don't know.
Perfect, great, thank you. All right, so next up, okay, how can patients or researchers be stewards in ensuring research and data are not siloed and are made as broadly available as possible to academic and industry researchers?
I think it goes back to the informed consent form. You got to make sure that what is it that you're signing, right? And that in your communication with your researcher, if your researcher tells you that they will be working on their own samples and that they will, the ownership goes completely to them. So make sure that you understand that. Make sure you understand what kind of data they will be sharing. Our own informed consent form has an opt-in for displaying your genetic information. Make sure that you understand what that means. If you are, and I have to say 99% of our submitters now are fine with this, but you have to know that your genetic data can be identifiable. So if you're okay with that, make sure that you understand that and say, yes, I'm fine with my genetic data to be displayed publicly. If you really are for a very sensitive population and you don't feel comfortable with that, then you have to make sure that you opt out of something like that. So I think it goes back to the informed consent and to the communication with your researcher.
Yeah, most informed consents will also say something about the fact that the data obtained from your samples may be published in a de-identified manner. So that's one thing. And the journals that accept these publications typically require that the data that is associated to the model be deposited in public databases. So it's sort of a two-pronged system that kind of ensures that the research is not kept by the researcher and not shared with the community. And depending on the field, I think most researchers are very happy sharing data and results because it helps cross-pollinate research projects. But it's a requirement on most journals now. So if you have RNA sequencing, DNA sequencing, you typically have to deposit that in the paper when you get it published.
Got it, thank you. And then it looks like we got 5 minutes left. So if anyone else has any other questions they would ask, please place that in the group chat box. We probably have time for 1 or 2 more. So I'll just start with one last question over here. What advice would you give to someone who finds the research process daunting?
My first advice is talk to other advocates. You know, your community is amazing. Find out other parents, other advocates, other patients that have gone through the process. Ask them who they work with, what recommendations they can give you, what mistakes they made, so you can improve on them. That's the best you can do. Also, talk to other people with your same disease. Talk to other people within your group. If you don't have a group, go and create one. If you go and find one, I'm sure there's one out there, but if there isn't, go and make it yourself. You're stronger in larger groups, and there's many of them, many of you that can work together.
Yeah. Great, thank you. All right, and then I guess since we have a little more time left, what excites you both most at this moment about rare disease research? What's going on? So what excites you both the most?
Well, for me, it's the tools. There's crazy new tools, both to generate models, but also to study patient samples. So we're doing a lot of single cell research, which is extremely exciting. And we're doing a lot of deep sequencing, which is also really cool because now we're learning new things. The coolest part of my job is associating phenotype to genotype and other molecular characteristic of the disease. So that type of computational integration is also something that's rapidly evolving, with machine learning and things like that. So all the new tech and tools that is coming out is really exciting. And I think we're making progress faster now than we were 10 years ago. So that's very encouraging.
That's awesome. That's so great to hear.
for me, it's actually the involvement of patients. I think that's a great step forward in the research for rare disease. It's the patients getting involved, getting to be more knowledgeable, and to push us to do better. So that's very exciting to me.
Perfect. Yeah. All right. Well, thank you both, Deborah and Hervé, so much for helping answer these questions. And thank you to all of you attendees for asking your questions as well. So next up on the agenda from 3:30 p.m. to 5:30 p.m. Eastern Time, we do have our 2020 Cure Accelerator Live for Rare Disease session. So if you would like to attend that, please head back into the virtual platform to the auditorium. Click on that center image and that will pop up the agenda for you to scroll down, find the Cure Accelerator Live session, and click that orange join button in the right. Thank you all again so much, and we hope you all enjoy the rest of the event.
Thank you very much.
Bye-bye.
Bye. Thank you.